Once you have cloned your gene, that is, you've been able to identify the DNA associated with that gene, and you've been able to make large numbers of copies of the DNA, you've been able to amplify the DNA. The next step is to characterize the DNA or the RNA corresponding to the gene, and to figure out how big it is, what is the sequence of the DNA, how the gene works, and so on. And one of the foundational techniques in molecular biology is gel electrophoresis. Most often, the DNA samples that we get are not pure. They're actually a mixture of different DNA molecules, and we would like to separate all these DNA molecules. And gel electrophoresis provides us a mechanism, a way of separating DNA molecules. And what you do in gel electrophoresis is that you make a gel with either agarose, the sugar agarose, or you can also use another type of gel called a polyacrylamide gel. And you make little wells in this gel in which you can pour your DNA or RNA sample. The other thing you do when you're making this gel is that you include a fluorescent a DNA binding dye. And what these fluorescent DNA binding dyes do is, and examples are ethidium bromide or picogreen. These dyes normally do not fluoresce, but if they're bound to DNA, they fluoresce. So if you take the gel and you expose it to ultraviolet light, if you were using ethidium bromide, the dye that is bound to the DNA will fluoresce in blue light, and you will be able to visualize or see the DNA. Now, the DNA is in the wells, and then you apply an electric field to this gel. So you have a negative terminal and you have a positive terminal. DNA's phosphate backbone is negatively charged. So DNA is a negatively charged molecule, and that will cause the DNA to move towards the positive electrode. The gel is made up of agarose molecules linked up to each other to kind of form a matrix, and you can imagine it as a cobweb. What this means is that if a DNA molecule is longer, then it will get tangled up in the gel matrix, or it'll get tangled up in this cobweb and will move slower. Whereas if the DNA molecule is shorter in length, then it'll move faster. And this difference in the speed of migration of the DNA molecules causes different DNA molecules to separate, with smaller molecules closer to the positive electrode and larger molecules closer to the negative electrode of the gel. And in DNA, since it's a polymer of A's, T's, G's, and C's, the length of the DNA is proportional to the molecular weight of the DNA, and this causes the DNA molecules to get separated according to their molecular weight, with heavier molecules at the top and lighter molecules at the bottom. Usually you include a marker, and what a marker is, a mixture of DNA molecules of known molecular weight, known lengths, and then you can read off the length of any particular band or DNA molecule by comparing it to the bands in the marker. So one thing the gel tells you is how long or how big a given DNA molecule is. But the gel also tells you how much of a particular DNA molecule you have. And that's because if you have less DNA in a particular band or less DNA of a particular size, then less of the fluorescent DNA dye will bind to it. Less ethidium bromide or less picogreen will bind to it. Since there's less dye, the fluorescence emitted from that band will be lower. And therefore, um, if you have more DNA in a particular band, you will have more dye and therefore more fluorescence. And therefore, by looking at the brightness of the band, you can also deduce the quantity of the DNA present, and you can also include standards of known amounts of DNA to compare with, to actually quantitate or determine how much DNA you have. So brighter means more DNA, and dimmer means less DNA. And you can use gel electrophoresis not just for DNA, but RNA and protein as well. The main difference between electrophoresis for DNA and that for RNA and protein is that RNA and protein can form secondary structures. And that means the migration, which is according to size, smaller moves faster and larger moves slower, is no longer does not any longer have a direct correspondence with molecular weight, because a large protein can fold up into a very compact shape, and the same with RNAs, where an RNA can be really long, but it can fold up into a very compact shape and migrate faster. And so you can carry out gel electrophoresis under denaturing conditions, in which you use urea or detergents to break up these hydrogen bonds and van der Waals interactions that give RNA or protein their secondary structure, so that they their linear size is proportional to their molecular weight. However, the rest of the principles remain the same. You can determine both the size, you can separate them by size, and then determine the size as well as the amount, the latter by looking at the brightness or the amount of fluorescence you get from a band in the gel. Now, even though in a gel you separate by size, if you have more than one DNA or RNA molecule that have roughly the same size, you will not be able to separate them, because the separation is only by size or molecular weight. In that situation, you can use another kind of set of techniques called blots. And what you do in a blot is just like when you are screening a genomic or a cDNA library, and you use a probe that is complementary, that is a DNA or RNA molecule that's complementary to the gene of interest or the molecule of interest, and this probe is radioactively labeled so that you can visualize wherever the probe is. You can use probes to pick out or isolate a particular DNA molecule, even if it has the same size as other DNA molecules. So if you are doing analyzing DNA using probes, then you are doing a southern blot. Whereas if you are analyzing RNA using probes, then you are doing a northern blot. Now, a southern blot begins with gel electrophoresis. And so you have a gel, and you size separate your DNA by putting them in the gel and applying an electric field. And so after electrophoresis, you get the DNA bands that are separated by size. And we keep in mind here that one band may have more than one DNA molecule inside it, because you could have two DNA molecules that have the same size but a different sequence. Now, what you do then is you make a sandwich of this gel with a paper towel on one end, a salt solution on the other end, and between the paper towel and the gel, you put a membrane, a nitrocellulose membrane. The water moves by capillary action from the salt solution to the paper towels, and thereby transfers the DNA from the gel to the nitrocellulose membrane. Now, the nitrocellulose membrane is peeled off and is baked in an oven to permanently link the DNA to the membrane. And then you can use radioactive probes and incubate the membrane with the radioactive probes. The radioactive probe will only bind to the DNA molecules that it is complementary to. So even if you have several DNA molecules of the same size, the radioactive molecule will only bind to the ones that are complementary to the probe and the target that you're looking for. And then you wash away the excess probe and then expose a photographic film to the membrane, and because of the radioactivity, you'll be able to see a pattern on the film corresponding to the places where the probe bound, and that means the bands that contain the DNA of interest. And if you are analyzing RNA in a northern blot, the process is very similar, except you may end up doing denaturing gel electrophoresis rather than native regular gel electrophoresis. And a similar set of techniques allows also the visualization of proteins, except instead of probes that are complementary to the DNA or RNA of interest, you use antibodies that are fluorescently labeled to visualize the protein, and that is called a Western blot. And you carry out denaturing electrophoresis in a polyacrylamide gel. So it's called SDS page for the detergent you use, which is sodium dodecyl sulfate for denaturing, and PAGE stands for polyacrylamide gel electrophoresis. And the only other difference from southern and northern blots is that you don't incubate the membrane with a probe, but with an antibody that's fluorescent, and then you can illuminate the membrane with with light, so that the antibody can fluoresce and you can visualize the protein. In all the blotting techniques, just like in gel electrophoresis, the locations of the bands tell you the size of the molecule, be it DNA, RNA, or protein, and the brightness of the bands tells you how much of DNA, RNA, or protein you have, depending on which technique you are employing. Next, another important technique for characterizing nucleic acids is to determine their sequence. So that's called sequencing. And if you are determining the sequence of A's, T's, G's, and C's in DNA, you are performing DNA sequencing. And just like PCR, DNA sequencing is based on carrying out a DNA synthesis in vitro. So carrying out DNA synthesis in a test tube. And normally the DNA synthesis is a polymerization reaction in which DNA polymerizes deoxynucleoside triphosphates. And so they have 3 phosphate groups, and they have their deoxynucleoside, so they lack the hydroxyl group on the second carbon of the ribose sugar, but they do have a hydroxyl group on the third carbon of the ribose sugar. And what this does is this allows a new base to come in to form a phosphodiester bond, and this allows the chain elongation to continue. Now you have a variant of deoxynucleoside triphosphates called dideoxynucleoside triphosphates. And dideoxy means that you don't have a hydroxyl group even on the third carbon. And if you incorporate a dideoxynucleoside triphosphate, so this molecule during chain elongation, this will terminate the DNA synthesis reaction, since as soon as you've incorporated a dideoxynucleoside triphosphate, and for short we will say ddNTP, whereas deoxynucleoside triphosphates are ddNTPs. Once you have incorporated a ddNTP, it lacks the hydroxyl group on the third carbon of the ribose, and therefore you cannot form a phosphodiester bond, and the DNA synthesis reaction will stop after the addition of a ddNTP. Now, to determine the sequence of DNA, you carry out a DNA synthesis reaction. And what you need in this reaction is, of course, the template, and that's the DNA molecule whose sequence you are trying to determine. Since DNA synthesis requires a primer, you need the primer to provide that hydroxyl group on the third carbon to form the first phosphodiester bond. You have to include a primer, and usually these primers are radioactive, so that the daughter strands that you build during DNA synthesis can be visualized by exposing an X-ray film. And of course, you need the DNA polymerase to carry out the synthesis. You need the DNTPs to perform the chain elongation to carry out the polymerization. And one extra thing that you add during DNA synthesis for sequencing is that you add dideoxynucleoside triphosphate. In this case, we are adding ddATP. Now, this ddATP is added at a concentration that is 1/100th of the concentration of the DNTPs. So even though you have these ddATPs, they are not very likely to get incorporated into growing daughter strand. Now, since the ddATPs are so rare, for the vast majority of the template molecules, the DNA synthesis reaction will proceed as normal. And so you will basically use base complementarity to add, to build a daughter molecule, and it will run all the way through. And the vast majority of a double-stranded DNA that you will get will look like this. It will be the full length. However, from time to time, you will have template reactions in which, instead of incorporating the DATP, you will by chance incorporate a ddATP wherever there is a thymine in the template molecule. So you could incorporate a ddATP here, or you could incorporate a ddATP here, or you could incorporate a ddATP here. As a result, you will get 4 types of double-stranded DNA molecules at the end of this DNA synthesis reaction. The vast majority is going to be this length, the full length. However, you will have a small amount of double-stranded molecules that are only this length, a small number that are this length, and another small number that are this length. And in the next step, we take this reaction mixture, which contains these 4 different types of double-stranded DNA molecules, and run it on a polyacrylamide gel. So here is the polyacrylamide gel, and recall that in a gel, these are larger fragments and these are smaller fragments. The convention is to always show a gel where the positive electrode is at the bottom and the negative electrode is at the top. And in this gel, we are not shown the full size double-stranded molecule. And so even though there are 4 types of double-stranded molecules, we only see 3 bands in the lane with the reaction where we used the dideoxyadenosine triphosphate. Now, this smallest band corresponds to this molecule, the next biggest band to this molecule, and the largest band to this molecule. And in a sequencing gel, the position of the band, or the size of the band, tells us what position a particular base pair is in, and which ddNTP we run the reaction with. And in this case, we ran it with ddATP, tells us the identity of the base pair. For example, we can say that this position in the daughter strand, you have an A. This position also has an A, and this position has an A. And so we're saying this is the daughter strand. Next, in the DNA sequencing, we repeat this reaction, the DNA synthesis reaction, but this time, instead of using ddATP, we use ddTTP as the terminating dideoxynucleoside triphosphates. And whichever bands we see tell us that the daughter strand must have T's in those locations. So there's a T here, a T over here, a T over here, and so on. Then we run a third reaction, this time using ddCTP as the terminator, and we can infer wherever we see the band that there must be a C in that position. And finally, we run a fourth reaction using ddGTP as the terminator, and in that lane we can infer for each band that there must be G's in the daughter strand. And since this side is smaller, this must be the 5' end of the daughter strand. Since you're starting from here, the 5' end, and this must be the 3' end of the daughter strand. And now we can determine the sequence of the template strand, the sequence that we are trying to determine, simply by complementing. Template, and obviously they're anti-parallel, so I have 5' at the top and 3' at the bottom, and the template strand will then read T T A G A C C C G A T A A G C C C G A. And in this case, in this example, we already knew the sequence of the template strand, and you can see that the sequence that we have inferred from the gel matches the sequence that we were given for the template strand. And that is how we can determine the DNA sequence of a molecule by carrying out a DNA synthesis reaction, or rather 4 DNA synthesis reactions, and including dideoxynucleoside triphosphates, which terminate the elongation depending on the identity of the base pair at each location.